General anatomy of the respiratory system. General anatomy of the respiratory system consists of a tube that divides into small branching tubes in the lungs. External nares, rightward arrow, nasal cavity, rightward arrow, nasaopharynx, rightward arrow, laryngopharynx, rightward arrow, larynx, rightward arrow, trachea, rightward arrow, primary bronchi, rightward arrow, lungs. Secondary bronchi, rightward arrow, tertiary bronchi, rightward arrow, bronchioles, rightward arrow, alveolar sacs, rightward arrow, alveoli. Lungs, cone-shaped organs located in the thoracic cavity. Thoracic cavity is lined with a body membrane called parietal pleura, while the surface of lungs is covered with visceral pleura. The thin space between the two pleural membranes is called pleural cavity, which is filled with a clear fluid called pleural fluid to minimize friction between the tissues and to provide surface tension in the pleural cavity. Water molecules in the pleural fluid allow the two pleural membranes to adhere to one another, to prevent collapsing of the lungs. Surfactant, a chemical substance called surfactant secreted by the lungs, also facilitate the surface tension. The histology along the respiratory tract changes. From the trachea to the tertiary bronchi, the tract is lined with ciliated pseudostratified columnar epithelium, smooth muscle, and cartilage rings. The bronchioles are lined with cuboidal epithelium, and from the alveolar ducts to the alveoli, the tract is lined with simple squamous epithelium. Inferior to the lungs is a sheet of skeletal muscle under diaphragm involuntary control, called, to facilitate the control of thoracic volume. The bronchial tree, like branching tubes extended from the trachea. Only the primary bronchi are external to the lungs, while the rest of the bronchial tree is embedded in lung tissues. Diameters of the tubes from primary bronchi to tertiary bronchi are large, so that support with cartilage rings is necessary. Diameter at the bronchioles is down to 1 mm, where the tubes do not need cartilage rings for support. This structure is composed of cuboidal cells where diffusion is also not possible. From the alveolar duct to the alveoli, the lining tissue becomes simple squamous epithelium where gas exchange is possible. Since there is a much larger surface area at the alveoli, almost all gas exchange occurs at the alveoli. 300 million alveoli provide a total surface area similar to a tennis court. Breathing mechanisms, physical laws. The gas law states that gas molecules always diffuse from a higher pressure area to a lower pressure area. The Boyle's law states that pressure and volume are inversely related, with the temperature remains constant, where pressure will increase in a smaller volume of gases, and pressure decreases in a larger volume of gases. Inspiration, inhalation, an active process where nerve impulses from medulla oblongata cause the contraction of diaphragm and external intercostals muscles. As these muscles contract, thoracic volume increases, which decreases the pressure within the lung, intra-alveolar pressure, due to the Boyle's law. When intra-alveolar pressure falls below the atmospheric pressure, 758 mmHg versus 760 mmHg, respectively, the gas law dictates that now gases move from the environment into lungs. Expiration, exhalation, a passive process where elastic tissues of the lungs and diaphragm recoil to their original position. As the diaphragm and external intercostals muscles relax and recoil, thoracic volume decreases, which raises the intra-alveolar pressure, again due to the Boyle's law. When intra-alveolar pressure is risen above the atmospheric pressure, 762 mmHg versus 760 mmHg, respectively, gases move from the lungs into the environment, again due to the gas law. Pulmonary ventilation. Inspiration. Pulmonary ventilation is the mechanism by which air is exchanged between the atmosphere and the alveoli. Air is exchanged due to the expansion and contraction of the lungs. Contraction of the diaphragm pulls down, enlarging the intrapleural cavity. Elevation of the ribs also expands the intrapleural cavity. These factors decrease the intrapleural cavity pressure, thus, air flows into the lungs. Inspiration, pulmonary ventilation. Expiration. During expiration the diaphragm relaxes, the ribs are pulled down. This increases the intrapleural cavity pressure. This results in the movement of air out of the lungs. Normal quiet breathing is accomplished entirely by the movement of the diaphragm. In the normal-sized person, about 6 liters of gas per minute move in and out of the lungs. Ventilation can increase up to almost 100 liters per minute during maximal exercise. Lung capacities, air volumes contained in the lungs, can be measured by a spirometer, and provide invaluable information regarding the normal function of the respiratory system. Tidal volume, TV, amount of air moving in and out of the lungs during normal breathing. Average value is about 500 ml. Inspiratory reserve volume, IRV, amount of air that can be inhaled after normal inspiration. Average value is about 3000 ml. Can be calculated by IRV equals IC, TV, where IC represents inspiratory capacity. Expiratory reserve volume, of, amount of air that can be exhaled after normal expiration. Average value is about 1100 milliliter. Inspiratory capacity, IC, total amount of air that can be inhaled. Average value is about 4000 milliliter. Can be calculated by IC equals VC, of where VC represents vital capacity. Vital capacity, VC, total amount of air that can be exhaled. Average value is about 5000 ml. Can be calculated by VC equals TV plus IRV plus of. Can also be determined by predicted VC values on a chart based on a person's age and height. Residual volume, RV, amount of air that is always left in the lungs after expiration. Average value is about 1200 ml. Total lung capacity, TLC, total amount of air that the lungs contain, including residual volume. Average value is about 6000 ml. Can be calculated by TLC equals VC plus RV. Anatomic dead space refers to the amount of air remain in the bronchial tree that is not involved in gas exchange, due to obstruction of airflow or damage in the bronchial tree. Alveolar dead space refers to the amount of air in the alveolar ducts or alveolar sacs that is not involved in gas exchange, due to poor blood flow or unusually long diffusion distances in gas exchange. Physiologic dead space refers to the total amount of air in the lungs that is not involved in gas exchange, i.e. anatomic dead space and alveolar dead space. Control of breathing. 4 major factors that affect normal breathing: stretching in the lungs and thoracic walls. O2 level in the blood. CO2 level in the blood. H and level in the blood. 5. Normal breathing is inhibited by stretching of the lungs and thoracic walls, a rise in O2 level, and a decrease in CO2 and H and levels, while normal breathing is stimulated by relaxing of the lungs and thoracic walls, a decrease in O2 level, and a rise in CO2 and H and levels. 6. Chemicals and emotional state also affect breathing. Stretch of tissues inhibits inspiration by triggering an inflation reflex which reduces the duration of inspiratory movements. This also prevents overinflation of the lungs during forceful breathing. Hyperventilation decreases carbon dioxide concentration as well. 7. Low blood PO2 increase alveolar ventilation. Peripheral chemoreceptors in the carotid bodies and aortic bodies detect low O2 concentrations. High blood PCO2 increase alveolar ventilation. High CSF, H and ion concentration increase breathing rate and alveolar ventilation. CO2 combines with water to form carbonic acid, which in turn releases H and ions in CSF. 9. Respiratory centers. Normal breathing is arrhythmic, involuntary action regulated by the respiratory centers in the pons and medulla oblongata of the brainstem. Rhythmicity area in the medulla oblongata sets the basic rhythm of inspiration and expiration, and is subdivided into the dorsal respiratory group, which controls normal breathing, and the ventral respiratory group, which controls forceful, voluntary breathing. 10. Pneumontaxic area in the pons sets the depth, duration, and rate of breathing by influencing the dorsal respiratory group. 11. Chemoreceptors. Central chemoreceptors are associated with the respiratory centers. CO2 combines with water to form carbonic acid, which in turn releases H and ions in CSF. Stimulation of these areas increases alveolar ventilation. Peripheral chemoreceptors are in the carotid bodies and aortic bodies. These chemoreceptors sense low O2 concentration. When O2 concentration is low, alveolar ventilation increases PCO2. Medullary chemoreceptors are sensitive to the pH of CSF. Diffusion of CO2 from the blood into CSF lowers the pH of CSF by forming carbonic acid. Similarly, the aortic and carotid bodies are stimulated by a fall in blood pH, induced by increases in blood CO2. pH. Peripheral chemoreceptors are stimulated by decreased blood pH independent of the effect of blood CO2. Chemoreceptors in the medulla are not affected by changes in blood pH because H and cannot cross the blood-brain barrier. PO2, low blood PO2 augments the chemoreceptor response to increases in blood PCO2 and can stimulate ventilation directly when the PO2 falls below 50 mmHg. Ventilation, the amount of air moved in and out of the lungs during each minute, is called pulmonary ventilation. Increase in metabolism is accompanied by increases in ventilation, due to increase in plasma CO2. Gas exchange. Respiratory membrane is formed by the walls of alveoli and capillaries, where they are both made of simple squamous epithelium, thin enough to allow diffusion of gases called gas exchange to occur. Dalton's law, gases, particularly O2 and CO2 for this discussion, always diffuse from high pressure to low pressure. Each gas in a mixture of gases produces its own pressure called partial pressure, pp or P, and the sum of all partial pressure is the total pressure of that gas mixture, a physical law called the Dalton's law. Therefore, the directions of diffusion during gas exchange in the lungs and in body tissues are based on the differences in partial pressure of these gases. External and internal respiration. External respiration occurs in the lungs to oxygenate the blood and remove CO2 from the deoxygenated blood. O2 diffuses from the alveoli into capillaries, while CO2 diffuses from the capillaries into alveoli. Internal respiration, tissue respiration, occurs in the body tissues to provide O2 to tissue cells and remove CO2 from the cells. O is critical in the release of energy molecules, i.e. ATP, is critical in the release of energy molecules, i.e. ATP, a process called cellular respiration, while CO2 is a byproduct of metabolism which can become harmful to tissue cells in large quantities. O2 diffuses from the capillaries into tissue cells, while CO2 diffuses from tissue cells into capillaries. Alveolar gas exchange. Gas exchanges between the air and the blood occur within the alveoli. 1. The alveoli are tiny sacs clustered at the distal ends of alveolar ducts. 2. The respiratory membrane consists of the alveolar and capillary walls. Gas exchange takes place through these walls. 3. Diffusion through the respiratory membrane. O2 diffuses from the alveolar air into the blood, CO2 diffuses from the blood into the alveolar air. Note, the differences in partial pressure determines the diffusion through the respiratory membrane. Pulmonary versus systemic capillaries. Pulmonary capillaries, alveolar PO2 equals 104 mmHg. Pulmonary capillaries PO2 equals 40 mmHg. Result, O2 enters capillaries. Alveolar PCO2 equals 40 mmHg. Pulmonary capillaries PCO2 equals 45 mmHg. Result, CO2 enters alveoli. Systemic capillaries PO2 equals 104 mmHg. Tissues PO2 equals greater than 40 mmHg. Result, O2 enters tissues. Systemic capillaries PCO2 equals 40 mmHg. Tissues PCO2 equals 45 mmHg. Result, CO2 enters capillary. Gas transport. 1. Oxygen. 98% of O2 is transported by binding to hemoglobin in erythrocytes. When O2 binds with hemoglobin, Hb, oxyhemoglobin, oxy-Hb, is formed, which shows are a red pigment. 2% of O2 is dissolved in the blood plasma. The resulting oxyhemoglobin is relatively unstable and releases its O2 in regions where PO2 is low. More O2 is released as the blood conc of CO2 increases, as the blood becomes more acidic, and as the blood TEMP increases. The efficiency of oxy-HB releasing O2 to tissue cells during internal respiration is shown on the O2-HB dissociation curve, which shows a distinctive sigmoid shape. On this curve, as O2 partial pressure increases, the level of HB saturation increases. Each HB molecule can bind up to 4 O2 molecules. At about 40 mmHg of O2, roughly 75% of HB is saturated. At about 80 mmHg of O2, close to 98% of HB is saturated, and the curve becomes flattened beyond this point, where only about 98 to 99% of HB can be saturated no matter how high the O2 pressure is. 5 main factors that can shift the O2-HB dissociation curve. 1. O2 pressure, a decrease shifts the curve to the right. 2. CO2 pressure, an increase shifts the curve to the right. 3. pH of blood, more acidic shifts the curve to the right. 4. Body temperature, an increase shifts the curve to the right. When this dissociation curve shifts to the right, it is a phenomenon called the Boer effect, where less HB saturation occurs and more O2 is being released from 5-oxyhemoglobin. When the dissociation curve shifts to the left, which is less common, it is called the Haldane effect, where more HB saturation occurs and less O2 release. Gas transport, carbon dioxide. 7% of CO2 is dissolved in the blood plasma. 23% of CO2 binds with hemoglobin in erythrocytes. When CO2 binds to HB, carbaminohemoglobin is formed, which shows a bluish pigment. 70% of CO2 reacts with water and forms carbonic acid in erythrocytes. CO2 plus H2O rightward arrow H2CO3. Erythrocytes CO2 plus H2O rightward arrow H2CO3. Carbonic acid is immediately broken down by the enzyme carbonic anhydrase, Ca, to become hydrogen ion and bicarbonate ion. H2CO3 rightward arrow H plus HCO3, where H end quickly binds with HB to prevent it from affecting blood pH too drastically, and HCO3 diffuses into blood plasma and maintains an ionic balance with chloride anion, Cl. Gas transport, carbon monoxide co-forms as a result of incomplete combustion of fuels. It combines with hemoglobin more readily than O2 and forms a stable compound. Copyright 2009. Ebneshahidi County is toxic because the hemoglobin with which it combines is no longer available for O2 transport. Clinical terms: anoxia, absence or a deficiency of O2 within tissues. Asphyxia, deficiency of O2 and excess of CO2 in the blood and tissues. Atelectasis, collapse of a lung or some portion of it. Bronchitis, inflammation of the bronchial lining. Cheney, strokes, respiration, irregular breathing pattern of a series of shallow breaths that increases in depth and rate, followed by breaths that decrease in depth and rate. Dyspnea, difficulty in breathing. Hyperoxia, excess oxygenation of the blood. Hypernia, increase in the depth and rate of breathing. Clinical terms: hypoxia, diminished availability of O2 in the tissues. Pneumothorax, entrance of air into the space between the pleural membrane, followed by collapse of the lung. Tachypnea, rapid, shallow breathing. Asthma, the dyspnea, wheezing and other symptoms of asthma are produced by obstruction of airflow through the bronchioles that occur in episodes or attacks. Obstruction is due to inflammation. Lung cancer, 1/3 of cancer death in the U.S. Smoking is the leading cause.